Yes, it's Sam Kelly guy. In this video, we're going to talk about the 4 substituted quinolines. And we're going to talk about specifically the chloroquine in this video. We're going to talk about the site of action, mechanism of action, resistance, how to recover this resistance, how to overcome the resistance, specific indications, talk about the salts, adverse drug reactions, and SAR. Please don't forget to subscribe, like the videos, and share. For chloroquine, this is the structure. This is the structure as you saw from the previous videos. That is chloroquine. For the site of action, as we saw in the first video, that chloroquine acts at the heterocytic stage, at the heterocytic zones, like quinine, and they have the same mechanism of action because it has a tertiary amine and a quinoline nucleus, or the quinoline ring. So they have the same mechanism of action as chloroquine. They prevent or they inhibit the metabolism of hematine, or the free hem to hemozoin. So this one leads to destruction of the parasite. So the parasite is destroyed, as we saw for chloroquine. I mean for quinine, this is chloroquine. Now for the resistance, remember this drug is basic. Here, the pKa is around 10.2 here. Then also here, it's around 8.4. As you say that this drug can enter, it is basic. When it enters the parasite, it goes in the food vacuole. And the food vacuole, the pH there is acidic. Means that this drug will be trapped. And when it is trapped, it will bind to the hematine, prevents its conversion to hemozoin. Now, how does resistance comes? For the resistance, the channels, remember, the channels where this drug penetrates into the food vacuole, they are positively charged. So, the mutation known, there are two mutations known, by the main one is the Plasmodium falciparum chloroquine resistant transporter. Transporter gene, or call it PFCRT. This gene, if there is mutation in this gene, if there is mutation, it will lead to change in the channel proteins. Now, the channel proteins, the positively charged will reduce. They will become, the positive charges of the channels will reduce, meaning that this drug, this channel can allow this drug to be pumped out. So the drug can move out. Because what was keeping there, because the channel positively charged, and also the drug is trapped because it is positively charged inside. So when the channels are made to be less positive, means that this drug can move out of the food vacuum, meaning that less drug is available to bind to the hematine. So that's one mechanism of resistance. Then another mechanism of resistance is due to the CYP450 enzymes. And the main one is CYP2D6 and also CYP3A4. These ones lead to the alkylation of the chain. Here at N, they will lead to loss of the chain. So we shall have hydrogen here instead of ethyl group. So there is the alkylation, means that the drug is inactivated. When the drug is inactivated, we may have resistance because there is reduced concentration of that drug to combat the plasmodium, and mainly for the plasmodium falciparum. So, mean that this species, the falciparum species, is very resistant. It can be resistant. It can get resistant to the drug due to this gene mutation and due to CYP450 enzymes. So, how to overcome this resistance is that this drug can be co-administered. Co-administration of the drug with the CYP450 inhibitors. The drugs which can inhibit these CYP450 prevent the alkylation, one of the resistance mechanism. So drugs like verapamil, phenothiazines, amlodipine, fluoxetine, and other drugs. Though this is not commonly practiced, but it's one of the way we can reduce the resistance for the chloroquine. Now, for the salts, for the indications, this drug, chloroquine, can be indicated for PVVAX, oval, malaria, for the malaria, and also for the P. falciparum, but for the susceptible strains. So that's the indication. And this drug, we have two types of prophylaxis. For prophylaxis, we have the casual one, the casual prophylaxis, and we have the suppressive prophylaxis. This one prevents the liver stage, the liver stages. This one, the casual one. But for the suppressive, this one eradicates the erythrocytic stages. And for chloroquine, it can be used for suppressive prophylaxis. Can be used for suppressive prophylaxis of these species. So that is for the indications. Now for the salts, we have the HCl salt, and also we have the phosphate salt. This one is mainly for parenteral administration, and for the phosphate, it's mainly for the oral administration. Those are tablets mainly. Those are the main salts we have: HCl salt and phosphate salt. Mean that if it is HCl, we shall have, here we had ethyl group. We shall have here that's the HCl salt. If it is phosphate, same thing. For the adverse drug reactions, for the ADRs, these drugs, they are not much selective. So chlorine, the chloroquine is not much selective. It can lead to hemolysis. I say that destruction of the red blood cells. These drugs, if they target the plasmodium, they can also lead to destruction of the red blood cells. So we can have hemolysis by the spleen. And this is especially in patients with glucose 6-phosphate dehydrogenase deficiency. And we saw this in the first video, how it can treat the red blood cells, the glucose 6-phosphate deficiency. This one can lead to hemolysis. And when you add the chloroquine, again, there is hemolysis, so it can worsen hemolysis. You can have retinopathy. Also, you can have muscular weakness, impaired liver function, also exacerbation of psoriasis and failure. So those are the ADRs for chloroquine. When it comes to the SAR, this nitrogen of the alkyl chain is for ion trapping into the food vacuole. So meaning that the drug can stay in the food vacuole and prevents the conversion of hematine or the free hem into hemozoin. It's the parasite is destroyed. Remember, it is basic. Then we have this quinoline ring. It's for also binding to the hemozoin, I mean the hematine, by stacking. The chlorine here, this one increases the affinity for binding to hematine or the free heme, and should be electron withdrawing. It is electron withdrawing group. Substitution of that chlorine, this one, with electron donating groups diminishes the activity. So it should be electron withdrawing group here. Here, this chain should be alkyl diamine. We should have these two nitrogen, this one and this one, with the alkyl group. So the linkage should be 2 to 5 carbon. This linkage between this nitrogen and this nitrogen. Less than 2, decreases the activity, and more than 5 increases toxicity. More than 5 increases toxicity. Also, here we saw that hydroxylation of the alkyl group at nitrogen here, hydroxylation, gives us hydroxychloroquine. And this one has reduced toxicity. Here. Though it reduces the potency, because we are reducing the lipophilicity, reduces the potency, but increases binding. We shall have binding by hydrogen bonding to the receptor here. Then we have this position here. This carbon here is chiral, and chirality provides us with the isomers. By these isomers, almost have same activity, antimalarial activity. So whether it's R or S, we shall have almost same activity. This part can be cyclized. We can form a cyclic ring here. But cyclization retains the activity. Also, the number of carbon atom for the chain here from this position up the end, the number of carbon of the side chain should be between 4 to 10. Between 4 to 10 for optimum activity. More than this will increase toxicity, and below this reduces activity. Below 4. Yes, for chloroquine, let's stop here. In case you have any question, please put it in the comment section. We shall answer it. Same the guy. Please don't forget to subscribe, like the videos, and share. Thanks so much.